Good evening. I'm Lynn Rubin, and I'm delighted to welcome you all to Genelia and tonight's Dialogues of Discovery lecture. Normally, my husband, Gerry Rubin, would be doing this, as well as introducing the speaker, but he is the speaker, so I get to do it. You all received a booklet, which I have one here, visual aid, that tells about Gerry's career in science. So I'm not going to go over that. I am going to tell you something about him that's not in there. So Gerry and I have been married for 38 years. And one thing that I can tell you, rock solid, is he works a lot. Pretty much all the time. And he's been like this for as long as I've known him. In fact, we met at work. I worked in a lab down the hall from his first lab at the Dana-Farber Cancer Center in Boston. And let's face it, where else was he going to meet someone? So I gradually came to realize that the reason Gerry works so much is he actually loves what he does. He's interested in it, and he's excited about it, right up until the point where he's not. So over the years, every few years, Gerry would start to get antsy. And he'd start looking around for some new scientific problem to solve. So to put that in terms we all can relate to, some of us watched Downton Abbey for years. We watched every episode, we cried on the closing credits of the last episode, and some of us are still in withdrawal, right? And then there were some of us who watched it maybe like till Anna got thrown in prison. And then we said, gee, maybe I should start watching Game of Thrones. Well, that would be Gerry, except it wouldn't be Game of Thrones. He'd be that guy who started watching Mr. Robot before any of the rest of us even knew what it was. Okay, so this pattern continued over the years. And in the spring of 1999, Gerry got on a plane and flew to Boulder to meet with Tom Check. And Tom had just been named the next president of the Howard Hughes Medical Institute, and he was trying to recruit Gerry to come work with him. So Gerry went to Boulder, he came home, he put down his suitcase, and he pulls a piece of paper out of his pocket. It's a paper napkin, and it's covered with numbers and drawings, and it's got holes in it because, you know, when that little bit that you're working on gets full, you open it up, right? And now you're writing on tissue paper, and it's probably the napkin that was under your drink, so it's damp. Okay, so this piece of paper looks like hieroglyphics. It's got holes in it. The ink is blurred, and he is spreading it out like it is some precious relic. So anyway, he starts talking a mile a minute about this idea that they have for a standalone research institute for HHMI. And his face is lit up like a Christmas tree. And I look at him, and I know in that moment, we are moving. So tonight's lecture is titled Genelia at 10. But you should know two things. One is that Gerry's been thinking about, talking about, planning, building, and directing Genelia for 17 years. And he's as excited about it today as he was when he pulled out that napkin.
Wow. Well, thank you. You know, in the 38 years we've been married, Lynn's always been part of my work. And many of her ideas on how to build a sense of community, how to build a family-friendly research environment permeate today at Genelia. And I've learned many things from Lynn. Early on in our relationship, she showed me the rewards that come from perseverance, of working hard for what you really want and not giving up. You see, after turning me down twice, she finally agreed in the third attempt to marry me. So, there you go. worth waiting for. I'm going to tell you about Genelia, the research campus. It's part of the Howard Hughes Medical Institute. The Howard Hughes Medical Institute was founded by Howard Hughes himself in 1953 with a very simple mission, shown here. Now, we're a charity, but we've only ever had one donor. But fortunately, he was very well off, and so we've been living on his gift for the last, 60 years. HHMI as an organization has always believed on supporting people, not projects. So most of HHMI's support for scientific research, even today, goes to supporting individuals called Howard Hughes Medical Institute investigators that are scattered around 50 or 60 universities and research institutes around the country. And in fact, I started as an employee of the Institute 30 years ago as one of these investigators in Berkeley. And this mechanism of supporting people worked very well. We found really talented people, we gave them generous support, and we stayed out of their way. But every 5 years, we'd judge whether they were worth more support or not. So this is a good mechanism, people, not projects. So why did HHMI decide, as we did in 2000, to build a freestanding campus? The existing model was working great. In fact, 13 HHMI scientists have won Nobel Prizes in the last 15 years. There's no other organization that can claim that. But at the same time, we knew from having worked in universities that there are limitations to a university environment. Our investigators were also faculty members. They had many other important responsibilities, including teaching and mentoring and running research labs. And the history of science taught us that certain kinds of breakthroughs really require concentration, ignoring the rest of the world, being in an environment where long-range research was supported, where there were no disciplinary boundaries, people worked together in collaborative ways. But many of these institutions had ceased to exist. Places like Bell Labs, they were industrial labs, and the pressures of the modern economy, looking at your quarterly earnings, drove those places sort of out of business. So we thought, I wonder if we can recreate what made those places so unusually productive in a new institution, starting from scratch on a nice farm in Northern Virginia. So that was the idea. I did my graduate work in one of our models, a place called the Medical Research Council Laboratory of Molecular Biology, which was in Cambridge, England. And one of my scientific heroes I met there, this kindly, or well, maybe nerdy looking guy, here, and I'm going to read you something that he wrote in his autobiographical essay. Said, "I've indeed actively tried to avoid both teaching and administrative work. I do not enjoy them, whereas I find research most enjoyable and rewarding. I managed to work in the laboratory as my main occupation from when I started as a PhD student until I retired. Unlike most of my scientific colleagues, I was not academically brilliant. However, when it came to research, where experiments were of paramount importance and fairly narrow specialization was helpful, I managed to hold my own." So this is kind of modest, and I think you don't realize how modest it is until you realize that this is one of the 3 individuals who actually won the Nobel Prize twice for different discoveries. So there aren't many Nobel Prize winners in US universities who are still working in the lab every day working on the second Nobel Prize. It's not what this environment supports. So we had these as inspirations, and we were very public about our view that we were trying to make an environment that was different for doing research. And this, because HHMI was a big organization in US science, caught the attention of people. And they were really intrigued by the idea that we were doing something which we're explicitly calling an experiment in sociology on how to best do science. So unlike most new research institutes, say set up a new cancer research institute or Neurobiology Research Institute. We were not established to solve a particular problem, but to change the way in which research was done. Okay? So this was set up, and by the way, our investment now is over $1.5 billion. So it's probably the world's biggest sociology experiment. Now, in the next part of my talk, I'm going to talk about the actual building of Genelia, because especially for those of you who are local, the construction of this building, I think, was really interesting. So HHMI bought this piece of land in 2000, and in 2001, they held an international competition to select an architect to design the campus. And the person that won that competition was this man, Raffaele Vignoli, and he worked together with the Institute's architect, a man named Bob McGee, who was an expert in laboratory design. And I was then director of planning for the campus, and we used to meet in these design meetings once every 2 weeks over 2 years to actually design the campus in a very unusual and collaborative process for construction. So most big construction projects, you would hire an architect, they would design the building, they would have construction plans drawn up, you'd put them out to competitive bid for contractors, and the contractors would bid on it, you hire a contractor, and they build the building. That's the way every building I'm familiar with on university campuses is built. We were very different, I think. We knew what we were getting ourselves into, perhaps. We first hired the contractor, which was Turner Construction. And then we said to the contractor, send us your best cost estimator. We want them in the design meetings. So this way, when Raffaele Vignoli came up with this great idea, very aesthetically pleasing, we could look at the estimator and say, how much? And he might say, 100,000, or he might say, 2 million, or he might say, I don't know how to build that. So in any case, this was very useful. So, and it was a just-in-time design process. I explicitly remember this time. I was called out of my temporary office. They were meeting in the office buildings up on the hill, into a room and said, we need to have a quick meeting. We have to make a quick decision. It's 4 o'clock in the afternoon, and we had to do some minor part of the construction decision. And they said, well, can we think about it over for a couple of days? He said, no, you don't understand. They're coming tomorrow morning to pour the concrete, and we still have to draw the plans after you make a decision. So that was how the campus was done, and there were times they poured the concrete before the plans were. Anyway, never mind. Anyway, we got the bill. So Vignoli won the competition because, unlike all the other internationally famous architects who competed, he had this vision. Rather than just building a building on the side of the hill, he had the idea of building the building into the hill as a terraced garden. And so these sketches I'm going to show you were all hand-drawn and painted by Vignoli and were part of his submission to the HHMI trustees in this competition. And this idea won him the competition. This is the manor house on the top of the hill. And then this turned into plans and turned into models. We had these wonderful complete scale models of the entire campus. This is a portion of one of them. And then that turned into the actual construction. This is a top-down view of the campus, and you can see the terrace down the hillside. So on every floor, from the roof to the third, second floor, and the ground floor where we are, you can go outside on the building. So it was an unusual design. He had many other interesting ideas. He had this idea that if you want people to cooperate and collaborate, you don't want them hiding in separate rooms. You want them to see one another. So he came up with this idea that you should have the external corridor, glass on both sides, and the labs would be in here, and you could look out that way. By being in the side of the hill, of course, we only had windows on one side of the building, and that's what this looks like. In the end, he decided that the glass should be curved. It was one of the first buildings done where instead of normal surveying tools, they use high-precision GPS to lay it out because everything is curved in this building. And you can see here's a lab, and here's the outside world. And then he had this idea that on these floors, we should have these gardens. I don't know why he called them tomato gardens, but we never planted tomatoes. We did have flowers. We preferred flowers. But this is outside looking into the lab. You see there's two glass walls. You're looking through the corridors in between. And then you can look into these pavilions that had the offices. And of course, we have all these internal spaces. Not everything we do can be done in the daylight. We have a lot of specialized research spaces behind the building, buried underground underneath the hillside. So we started the construction in 2000 while we're at the same time making the plans. And so this is where you are now, and this was December of 2002. And then in early 2003, we're in the throes of the major part of the excavation. A lot of dynamite, a lot of rock. In fact, we had a rock crushing plant on campus where we made 100,000 cubic yards of gravel from the rock we took out. And then the thing began to take positive forms as 2004 and went on. And now by mid-2005, it sort of looks like itself. It took 3 million man-hours of work to build this campus. It was a major construction project. This is what the campus looks like today. You're sitting in an auditorium about right over here in the middle of the building, underneath here, and you probably saw there's a concrete wall when you look across from the front door. On the other side of that wall is a 96-room guest house. You can see the little beds poking out the windows there and how the building's terraced. So my favorite part of the construction, I can't tell you, I have lots of construction stories, and this was one of the most enjoyable times of my life with this, working with it's much more fun than science, actually. But anyway, for me, I don't know. For some of my colleagues will like looking at me like, never mind. I really like the way they put in the glass. You see they have these like cranes with little like suction cups, and you can see that, going in. And this is real structural glass. These are glass beams on glass walls holding up a glass roof. So this building has one of the largest amounts of structural glass of any building in the world. And the green roofs at the time we finished were the second largest green roofs in the United States. Okay, so now we had a building under construction. We had to talk about management structure. So everything we did went down to a clean slate. We thought, how should we run this place? How should it be managed? And now I want to quote from another of my scientific heroes, another person who I met when I was a graduate student. He was the first director and was still director when I was a student of this laboratory. He was a Nobel Prize winning scientist himself, and he was an extraordinary scout for talent. So you've heard of Watson and Crick and the double helix? He hired them both. They worked for him. They were in his laboratory. In fact, Crick was his graduate student. Watson was his postdoc when they did the double helix work. So he had enormous talent, and he also had a sense for what it takes to manage a research institute. And I'm going to quote from him again. Creativity in science, as in the arts, cannot be organized. It arises spontaneously from individual talent. Well-run laboratories can foster it, but hierarchical organizations, inflexible bureaucratic rules, and mounds of feudal paperwork can kill it. Discoveries cannot be planned. They pop up like puck in unexpected places. So this attitude was very powerful. And I have many other management influences. One of my other favorite management is this man, which I'm happy. Some of you, because we're in Ashburn, Virginia, don't like this man, but I actually think he is one of my other management heroes. We can see, anytime you talk about someone that people know, there are people that like them and don't like them, but that's the way it is. Okay, so we really tried to social engineer an environment. And social engineering has a negative connotation to many people, but not to me. We really wanted to make an environment, explicit environment, that we thought would be this combination of being somewhat anarchistic, but collaborative. And we felt this is what was needed to be successful. So we set out to engineer this, and again, a lot of people found this was strange, but that was our goal. And so we did a lot of things. I can't go through them all. They're all amateur sociologists trying to do this. But for example, this is our dining room that's down the hall, and this is a typical lunch hour, and it really is from 11:30 to 1. Everybody has to eat then if they want to get fed. And at that point, especially when we opened 10 years ago, there weren't a lot of other places to go near here to eat. And we had very good subsidized food, and people all ate, and they mingled, and they ate from different areas together. Coffee. People like coffee. We decided, I don't know about this water cooler thing, I mean, I never got that because people aren't always that thirsty, but scientists like coffee. And we decided if we give free coffee one place in the building, this building is a sixth of a mile long, it can take 5 minutes to walk, from your lab to the coffee. But if the coffee's good and its coffee is free, people will come there. And I often just hang around there for 15 minutes a day, and it's much better than email for doing a lot of business. So this is very popular. To give you an idea, we have free coffee and free tea. We have it 7 days a week from early in the morning till late in the evening. We go through 2,000 coffee cups a day. So that's 500,000 coffee cups, the coffee or tea, a year. It really works, people. You can attract people with food or beverage. We also have on-site childcare, a number of other a fitness center, things to try to make this a good environment. All right, so up to now, we've been talking about a scientific research campus, and I haven't said anything about science. But I want to remind you that our primary goal was to set up a place to do science in a different way. And up until about this point, we didn't really give a lot of thought to what are the research problems we're going to work on? What are we going to study here? But then we realized when we were about at this stage, the building was half built. We said, pretty soon we're going to have scientists work here. We better decide what kind of scientists to hire and what to work on. So from our general concepts of thinking about it, we realized, those of us who were involved in our advisors, we have lots of committees, advisory committees, that in universities had a lot of strengths, but one of the things they weren't typically good in, nor was the federal government's funding mechanisms, was supporting people who wanted to build new research tools, new microscopes, new other apparatus, new research biochemical tools, because it was just underappreciated. And we also knew, and because of this, universities didn't hire the right kind of faculty to want to do that kind of research. At the same time, even when universities tried to do it, they had this problem that the engineers and physicists were in one department and the biologists were in another department. And we also felt that you couldn't build tools that way. You had to have the tool builder next to the person using it so they could tell them whether their tool worked or not, or what the tool was, or what the problem was, and could tell them when they had something, whether it worked or not, in a very rapid way, not by reading each other's papers 6 months later. So we came up, this is our initial mission statement, was to look for areas where limitations were technological, then hire tool builders and biologists to work together on those problems. Of course, they had to be important problems in the biomedical area. So that's what we started out with. And then we had a whole series of planning workshops to consider different areas that were suggested, and we accepted some and rejected others. And we ended up with these two areas. The first is the general mechanism by which brains work, if I can say that. So computers compute things, and they form calculations, and they do a lot of things, and with artificial intelligence, they're trying to be more like human brains. And we sort of have some idea of how computers do what they do because humans designed them and humans wrote the software. So we have at least some pretty good understanding of what's going on. But we have almost very little, none, I wouldn't say that because it's always there. We have very little relative understanding of how computational devices that were designed by evolution work. They're very powerful. They're much more energy efficient and much more powerful than any man-made computer. We like to understand how they work at a very basic level, so we study them in very simple animals like flies and fish and mice, figuring what we learn will be relevant for what happens in all human brains. So that's one of our research areas. The other area was to develop imaging and image analysis technologies. And these areas are synergistic, and I'm really going to focus, because there's more pretty pictures in the second one than the first one, on the second area. But I need to explain to you why imaging is so important. So we have to do a thought experiment. Imagine you've arrived here from a foreign country, foreign planet, say. And you've never heard of any of the sports games that Earthlings play. And someone showed you a set of pictures. They showed you that picture, that picture, that picture. And they said, what is this? What are they doing? And what are the rules of this game? Now, the pictures are certainly helpful. And in fact, a lot of the work we did in imaging was making better microscopes to take more detailed, higher resolution, clearer pictures like this, snapshots. But it's hard from these snapshots to actually figure out the rules of the game. Now, if instead of the snapshots, I show you a video. Whoop, that was the wrong one. This video clip. You can sort of get an idea that there's this funny shaped ball and they're throwing it and people fighting over who gets to catch it. So this is what a movie gives you. You already have a lot more information. So we're trying to build instruments that can take movies like that of live cells, developing embryos, which neurons are active in brains when animals are performing a task. We'd like to do that. And then from that and putting a lot of other methods, we want to get a little more insight that might be like this video where it's narrated.
Pass is intercepted at the goal line by Malcolm Butler. Unreal.
So now you're getting a little better idea of what happened and why it was important and why these guys were fighting over the ball. So that is in a way we're trying to look at these processes and record them in movies, and then add that annotation by doing other kinds of experiments. So now, so we had our areas, of course, but we had no scientists, right? So we had to hire the people. So in a typical university, you have faculty, and faculty have search committees, and people apply, and you interview the candidates, and you select who you want to hire. We had a construction site. A lot of good construction workers, but I don't think they would have been helpful in picking people. So fortunately, we had a lot of talented people. Remember I mentioned these Hughes investigators. And we got these Hughes investigators in 2005, a year before we opened. We're there, and this is the person who's going to come up here and give his talk, and to this kindly group of people. And actually, these people were pretty tough committee. You can see that many of them were highly qualified, and this was the then president of Howard Hughes. This is the person that went on to be his successor, and my new boss, Aaron O'Shea, who took over a month ago. So the people who were then president and next two presidents of Howard Hughes were in there, and people would give their talk, and then we would select and hire people. So I'm not going to talk a lot about the quality of the people we've hired. I'm very proud of them, but like my kids, I'm sort of like, it wouldn't matter what they were. I'd still be proud of them. But I can give you some metric. And so this is sort of like the National Academy of Science, to keep the sports analogy, is like the Hall of Fame for sports. They're people that their peers have elected as being distinguished scientists. And if you compare Genelia in our 10 years to other Virginia institutions, we do pretty well in the number of people that receive this honor that work here. So, all right, now I'm going to switch into the science that we do here. And in this part of the talk, I'm going to use a lot of video clips because I really prefer to let people tell you about their work in their own voice rather than me just telling you a whole bunch of scientific stories. And fortunately, we have a lot of these. Some of them, like this one, come from the PBS NewsHour, did a short piece on us, and I'm going to show you some video clips from this.
To pursue a greater understanding of neuroscience, Genelia scientists and engineers work in close collaboration to build new tools of discovery. Developmental biologist and neuroscientist Philip Keller showed me his amazing light sheet microscope.
So this is a setup that's designed to basically minimally interfere with the normal development, the normal function of the biological sample.
Living samples are bathed in laser light and recorded by 2 fast digital cameras to generate 3-dimensional movies of biological processes. This is a Drosophila embryo from 3 hours after fertilization until it hatches 24 hours later.
So every single little blob in this movie is one cell. We have tens of thousands of cells at this point in development. You can in fact now see these rapid movements. These are muscle contractions. So
this is one embryo from two angles, right?
This is exact.
Okay, so those were examples of that was a fruit fly embryo, and the other one, the one around at the beginning, was a fish embryo. And those embryos develop outside the body like a chicken egg does. And Philip's lab is also studying embryos that develop, vertebrate embryos that develop, like a mouse embryo that develop inside their mother's body. And for this, they had to make this slightly more complicated microscope because they had to provide the kind of environment that mimics the womb for these embryos so they could survive long enough to image that. So this is just a picture of one of these microscopes, and what you're going to see here is the stainless steel womb here that the little mouse embryo is going to live in and where it will develop for 2 days. And this is a movie taken by Kate in Philip's lab, and this is in time and hours. It's sped up a lot. And this is going to go, this is a mouse embryo going from about 1,000 cells to about 40,000 cells in 2 days. And you see it's more cells, it's taking some form here. At the end here, you can see this movement here. That's the embryonic heart beginning to beat. No one had ever seen a vertebrate embryo at this stage actually develop like this until they built this instrument. And of course, we want to look also at individual cells. So here's a movie from Eric Betzig's lab. And here, these orange cells are what's called T cells. They're part of your immune system. And they're attacking this blue cell. This is just the T cells alone from the same movie. And you see they're going after them. And again, this is a movie that runs in seconds. So it's sped up as well. So T cells have gotten a lot of good press lately because you've probably all heard of all the progress in cancer therapy, immunotherapy. And that works by getting your body's T cells to attack tumors. So it's these orange cells that are the good guys in cancer immunotherapy. And it's from studying them in this kind of detail we get a better idea of how they might work. All right, so now we can use the same kind of microscopes to work on our other problem because we'd like to understand how the brain works. What better way to understand how the brain works than looking at what cells in the brain are active when you're thinking or doing something. And so you're probably used in humans to use these big machines called functional magnetic resonance imaging, fMRI, and they say, oh, this part of the brain is lighting up when a French speaker speaks French, and this part of the brain is lighting up when an English speaker speaks French, or when you're upset, or whatever. But those are very low resolution methods. They have an area of the brain that's tens of thousands of cells. Using these kind of microscopes and sensors that were developed here at Genelia, proteins that change in their properties. They give off light when the nerve cells are active. You can view the brain when it's active, which cells are active. So this is another
There's
part of the
something
same.
fishy in Keller's lab as well. Zebrafish, here waiting for their close-up, an extreme close-up. Keller's groundbreaking movie shows a zebrafish larva brain in action. Each of these blips is a single neuron lighting up.
Whoa, wait, what happened there just now, that big flash?
We don't
It's like a thunderstorm.
quite know what the zebrafish was thinking at that point. But these types of experiments are aimed at trying to find out what exactly it is that
He was
is
thinking about
happening.
something good or something really
Hopefully.
bad, right? It was one or the other,
We
right?
tried our best to make sure that it doesn't have to think about something bad.
Okay. So this was the first time anyone had taken the entire brain of any animal and imaged all the cells at one time. Okay.
Whether they are
So the mouse brain is much bigger. And so you can't use this particular instrument for that. And so there had to be a special microscope built for that. And I'm going to show you a video that describes how a microscope is designed and built. And this is a video that's off our website.
The key projects in the lab is to build a new microscope. What we're trying to do with this new microscope is to image large parts of the brain, but with single neuron resolution. So not give up on the micro and not give up on the macro. So really what we want to do is to have our cake and eat it too. It turns out there's no reason, optical reason, why that can't be done.
We just made this piece.
Tanya is really the manager of the engineering group. She has a lot of technical know-how, especially in the area of mechanical design. Yeah, it's a complicated piece, yeah. We've been developing a microscope that allows us to do microscopy at the level of single neurons, but essentially over fields of view that are matched to at least the mouse brain, 5 millimeters or so. Could you pull this apart in distance?
Stretching this out another 5 inches, I mean, maybe that's possible, but it's going to make these objects way bigger. Figuring out how to do the mechanics of the transmission dichroa is kind of tough.
This has been a very challenging project. There's optical design that comes into it, optical physics. We're converging on a design and moving into a phase that is called tolerancing and manufacturing. Tolerancing is effectively wiggling all these elements and seeing how quickly performance degrades with Dan's model. And if that's catastrophic for many elements, then it will be very difficult to manufacture. Right? And we have no idea how that converts into cost. We'll have to come to a decision whether or not to go ahead with manufacturing. But it is still only a computer model that tells us that in principle it can be made, but the major outstanding issue, how well can it actually be manufactured?
So this microscope, it turns out, actually was made about a year after this video. And this shows some work done by, that was Kyle Zavoda, one of our group leaders, a graduate student in his lab, Nick. And what you can see, this is a big chunk of a mouse brain, about a quarter of the mouse brain. And these are individual small areas that you can zoom into. You can image the whole brain without zooming in these. And these are individual neurons, brain cells. And what you're seeing here is the ability to look at neurons in these different parts of the brain and monitor them and see which ones are active at the same time, that what might be connected and might work together. This is what this microscope looks like in a schematic. It's big. It weighs 250 pounds. This diagram is pretty much scaled. This is a mouse here. And the parts to build it cost about $250,000. So it's not something that you would necessarily go home and build in your garage after the lecture. But if you wanted the plans, we would give you the plans, the CAD diagrams, and all the software. So one of the issues for us is how we disseminate the tools we devise here to scientists all over the world. And we have two mechanisms. One, we license the design to a company who will make this and sell it. They won't sell a lot of them, but they'll sell enough of them to the researchers all over the world. And we also held a course here where we brought people from all over the US, from Portugal, China, Austria, Switzerland here to get a how to build your own lecture. And these people went home and built their own microscopes. And that gets the technology out to a dozen labs, and then other labs will be able to do this. We're very big, since we're tool builders, in making sure that other people can use the tools we develop. And I'm going to end the science part of this presentation with this last clip from
Researchers
CBS.
here are keeping long-term basic research afloat at a time when it finds very few safe harbors.
It can be a tough sell because, of course, not every avenue of research works out. You don't know exactly where it's going, and you don't know exactly what you're going to learn. That is the beauty of it. We don't know what the truth is, or we wouldn't need to pursue it. So sometimes it turns out as you expect, sometimes it doesn't.
There are no guarantees the research at Genelia will pay off with new Nobel-class discoveries. But that's precisely the idea. The sure thing need not apply.
So this is sort of prophetic because this was aired in July 2014. In December 2014, Eric Betzig, one of our first scientists, won a Nobel Prize for building a novel microscope. This is him and the King of Sweden. Okay, so I want to end my talk by talking about citizenship, and we're citizens of two communities. We're citizens of the international scientific community, and we're citizens of Loudoun County, Virginia. We serve the international community by doing science, by building research tools, by distributing them and making them available, and also by holding conferences. So we've held over 230 meetings, many of them in this room, more than 16,000 participants from 45 different countries have come here over the last 10 years. They are from early career stage scientists to very distinguished individuals. So this is one of the ways we give back to the community. And in the local community, we've tried to do a number of things to be good citizens of our community, supporting the local educational system and other things that are listed on this slide. And of course, the people that work here as individuals also make contributions. 400 of our 600 employees live in Loudoun County, and they do many things to support the local environment here. And of course, we hold these public lectures, of which many of them have been held here. We probably had about 15 of them so far on various areas of science. And when we have our lecturers come in, we try to get them to talk to local students. So this was a couple of years ago when we had Jennifer Torches come in, who's a very successful female computer science, mathematics whiz, who runs Microsoft Research in Boston, New York, come in and talk to some young women from Loudoun County high schools who were interested in potential careers in science and what it was like to be there. That we left them alone in a room. I mean, I was there for like 5 minutes, and I knew I had to get out because Jennifer was commenting about this young lady's hair and saying, yeah, when I was your age, I used to dress in leather and chains. And then I thought, I'm getting out of here, and these guys, young women, can talk about life. So we've done that. And this is where we are today. We've been open for 10 years. We hope to be around in this community for a long time. And I'd be happy to answer any questions about our past, present, or future. We're actually in Ashburn. Do you ever try to buy this amount of land in any of those cities you mentioned? No, seriously. Our headquarters is in Chevy Chase, Maryland, not far from the NIH. And our trustees felt it would be a good idea if they were going to invest all this money to be close enough that they could keep a good eye out on us. And that we could share infrastructure with headquarters infrastructure. And so our original search image was to be within an hour drive, which is still possible. Like now you could drive there in an hour. It's different, in 2000 when they were looking for that. And we wanted to be a place where we could be near where people would get here, so near an international airport. And we wanted enough land that we wouldn't run out of land, and we wanted to be already zoned for research and development, so that we'd be welcome in the community, and we wouldn't, in a lot of those places, you want to build a new research thing, and they would say, not in my backyard. So we wanted to be in a place that was all those things. And they hired a property firm to find such a site, and they came back and said, there is no such site. But there's this Dutch software company who's going belly up, those after the .com, and they had built these buildings here as their international headquarters. And so we anonymously approached them and said, what if we just make you whole, and we pay you what you paid for the land, and we pay you what you paid to build those buildings, and we'll take over. And that's how this land was selected. And then 6 years after that, we bought Selden Island, which is actually in Maryland. It's a large 470-acre island in the Potomac River, which is underwater a couple times a year, so you can't build anything on it, but it's great for running trails and for recreation. And the main campus is 270 acres, of which 160 is buildable. So that's the property story.
I have a very basic question that I'm hoping you can give a very basic answer to. In the end, what's the end goal? That's what I always wondered about Genelia. So after all this scientific research, you hope to do, like, you know, move cures for diseases forward, and it was referenced a little bit in that that's part of it. You just do and you research, but what's the end goal?
Fine.
In very general public language.
Yes, ma'am. You sound like my third grade teacher. Actually, no. All right, so we are the Howard Hughes Medical Institute. So everything that we do at Genelia, I believe, is medically relevant. It's just a matter of time. So if you were to go to the National Institute of Health and ask the people who are running the research there, they say, we want to cure Alzheimer's, stroke, Parkinson's, cancer, whatever, and we want to cure it before the next election, because that's what Congress wants us to do. This is not very practical. And, you know, for science, the history of science is that we build on prior discoveries. And so a lot of what we do now is only possible because of some science that was done 10, 20, 50 years ago by people not realizing that it would be relevant to this real world problem. So if I were to answer the question is, my lab studies the brains of fruit flies. And I think what we find out is going to be very useful for solving some of these neurological diseases I mentioned. And I'm very comfortable with that. But he said, well, when will that happen? I said, when my 7-year-old granddaughter is old enough to have Alzheimer's, I hope what we're doing today here will have led through a series of steps to cures. So we just have a very long time horizon to get the basic knowledge that will empower these studies. And I think that many of the tools that we built here, like the example of those movies that have the flashes of light in the neurons, they depend on a molecular tool that changes in its properties when neurons are active. And that was developed here, and over 1,000 laboratories all over the world are using that. And some of those people are doing research in very clinically relevant today. So a lot of the tools we do are disseminated to many people, but we have tended to focus on the long range, very basic research. We want everything to be medically relevant, but we're quite happy if it's 50 years from now that it's relevant. Is that okay?
I was wondering, when you develop something like the microscope you talked about, do you get a patent on that, or is it?
Yeah, we do patent our discoveries, but our main goal in licensing them and how we use those patents is to achieve our overall goal as defined by Howard Hughes, to increase basic knowledge for the benefit of mankind. So some of our new microscopes you can buy from Zeiss Microscopy Company, the biggest microscope building company, because they license the patent and they're now selling them. That's important for us because we can't make 1,000 of something and get it in different laboratories. But we have a policy of sharing everything. So all those microscopes, even the ones that have been exclusively licensed to a company, we will give anyone who just signs an agreement and says, I'm going to use this for my own research. I'm not going to sell it to people or rent it to people. I'll use it in my lab. We'll give them all the construction diagrams, all the software to run it, and they can build their own. So even though the microscope is exclusively licensed to a company, there'll probably be 50 people who build their own beforehand. And the companies like this because they're building a market and they're showing how useful it can be. So we just try to do whatever we can that other people can use our discoveries. Sometimes that includes licensing to a company.
I want to follow up on your I've been around this area for a long time. And one of the things about NIH was used to be sort of a sandbox. If you try to find a scientist who is working on stroke, they may be in pediatrics, and you couldn't figure out why they were there. And the reason was it was a sandbox. You discovered something, and it may not be relevant to the institute you were in. Do you try to model the way NIH used to be, not the way it's so focused now, so that if someone finds an area, you just let them dig into it, and wherever they end up is great? Or is it more focused?
Well, we do follow how, I would say that during my career, I never would have thought NIH intramural program thing in Bethesda as a kind of positive model. Although there are a lot of good sciences, but then they had a lot of good sciences. But it's a government institution that's very hierarchical and organized in a way. Maybe many years ago, it was like that. But we tend to follow the HHMI mantra of people, not projects, hiring good people. But on the other hand, we set areas we wanted to do when we said we wanted to get the people that need to come together. So in another way, you could look at it, we were a startup biotech company whose product was new microscopes, or a startup biotech company whose product was to understand the basic principles of the brain. And if you were running a company, you might say, I need an engineer, I need a software developer, I need an optics expert. And you would go and try to find people who knew those things, who wanted to work together. So it's selecting them because they said, wow, you want to support this? I want to do that. And I want to have other colleagues. I can even do something I could never do in my own lab. So that's, for a fraction of scientists, that's a very strong selector. And there were people when we started on our work on the nervous system who really wanted to work on the brain in this way, and it was very difficult to get money from the government then. Now we have the Brain Initiative from the government, and that is very closely modeled in its goals over what we started in 10 years ago when it wasn't popular. So I think the idea is you create a culture and a goal and a way of wanting to work together, and that's going to appeal to some fraction of people, and those people want to come here and work together. And that's, I think, been very successful for us. And then we have people that leave here and go off to great jobs elsewhere, and other people that come in.
Dr. Rubin, you've talked about how you hired scientists, but I believe you have lots and lots of young people working here, research fellows, interns, and from lots of different countries. And how many are there, and what countries are they from, and what skills do you look for in your young people who come here? Fortunately, I have lots of people who will correct me when my memory of these precise numbers are wrong in the audience, so please do if you think I got it wrong. I think we have about 500 employees, probably about 200, 250 PhDs, and they're about close to a third to a half of them are coming from all over the world. In other words, they're not from US. A third are here who come from all the world, and I don't know how many countries, 30 countries, I don't know. Yeah, a very large number of countries, and they come here for training, and sometimes they stay in this country, and sometimes they go back to their country. We have a lot of people. We have 25, we don't give degrees, but we probably have 25 people here who are doing their PhD graduate research, where they're doing their research here, and they're getting their degree. We have people getting their degree from the University of Chicago, from Cambridge University, from Hopkins, at least, and there are more, but I don't remember them, who come here to do their research. So we like to have people come in and mingle and spend part of their career here, and then they often move on. Science is a very international activity with a lot of cooperation and interaction, and so we fit into that mold. I don't know if that, and I didn't have time to talk about all the people we hired. Of course, we hired a lot of people, and I mentioned 400 of them live here locally. So makes us in the top, just in the top 50 employers in Loudoun County.
What is your biggest challenge going forward?
Well, you know, if I look at the organizations that I said were modeled after, they went through their heyday when they were the best. Everyone agrees Bell Labs was the best solid-state physics research lab for a while, but that doesn't really exist anymore, Bell Labs. And the MRC LMB also had a period when it was clearly the best in the world. And they're still good, they still exist, but they're not so unique anymore. So I feel like we've established a lot of momentum. We're really going in a great direction. We're reaching the top of our game. And how do we stay there? How do we keep, not get self-satisfied, not get complacent, keep challenging ourselves, getting out of our comfort zone? And how do you do that? And that's really, you know, a lot of scientists are basically, I would describe myself as being compulsive, obsessive, driven internally. As my wife mentioned, she doesn't have to tell me get up and go to work, stop lying around in bed. You know, this is what I want to do. When I get out of bed, I want to go to work. This is what we have to get, this excitement in people who really love what they're doing and want to keep going. So keeping the momentum going in the face of success. Because once you're successful, you build up expectation, right? And we're in a field of science which is definitely, what have you done for me lately? Or what are you going to do for me tomorrow? And past achievement means very little and shouldn't mean a lot, right? In terms of getting return on your investment. So that certainly is what I worry about. I mean, because I used to worry about, will we ever get the building built? Could we ever get people to move here from San Diego and Seattle and Princeton and Boston to come here? And I can tell you, we did get people from all those places. We got people from who were tenured faculty members at places like Princeton, Brandeis, University of Washington, UCSF, to give up their tenured jobs and come here for untenured jobs. So I was worried about that. Could we get them to come to suburban Northern Virginia? We proved we could do that. Could we get them to do good work together and do great science? I think we've done that. So if my worries change, I'd like to think I'm not really obsessive because I change what I worry about. And someone introduced himself as a psychiatrist, so I may get some correction. But I think it's always a challenge, and that's what makes my job, as my wife mentioned. It's a new challenge every day. There's a new problem to solve. We have, you know, what we do, in my mind, we're a luxury of society, the way the arts are. We get to do what we want to do. We get to do research. We have to give back. We hope to give back by making discoveries. But I worry every day, are we doing enough? And are we doing the right things? Great. It's 8 o'clock, and that was a great question to answer.